Good evening, people. Can you hear me clearly? Um, Good evening, everyone. Am I audible? Say yes or no. Am I audible? Rishma, good evening. Yes, yes. Right. Nice. Right. People, um, we have discussed the theory part related with the paper number 4. Paper 4, see if they have the paper number 4 with you. Paper 4. Right. People, paper 4 based on some sections. Paper 4 based on a few sections. What are the sections in paper number 4? Inorganic reaction, exergonic reaction. Um, I think it's based on part of chemical basis. What is phosphorylation? Enzyme lesson, and enzyme, energy relationship part, the enzyme lesson. Due to organelles carry out detoxification, cell structure function. And then people, cell cycle, small part is there from cell cycle lesson. And then people from photosynthesis. Second question based on photosynthesis, and then last part based on cellular respiration. Cellular respiration, right? Cellular respiration. Right. So we'll start with. No, we are not going to discuss it. Right? We're not going to discuss it. I'll upload the paper to the chat line now. I'll upload the paper to the chat line. Just give me a second for that. We'll discuss people the theory part relate with this. Maybe we can start with photosynthesis. Photosynthesis and enzymes. Those two parts will go through a little bit. Right, photosynthesis. So this is the paper. You heard the news, that there is another tropical depression is on the way, so reaching to us tomorrow. Anyway, hope there won't be a serious issue, right? So people will talk about this photosynthesis part, light dependent reaction. We can start from there, light dependent reaction, right? Light dependent reaction. So we talk about people, the photosynthesis has 2 steps. And the 2 steps are there, light dependent reaction and the light dependent reaction and the Kelvin cycle. Light dependent reaction and Kelvin cycle. So we learn the definition. Photosynthesis, metabolic process by which light energy is trapped and converted to chemical energy. We talk about people, the global importance and all. So go through a little bit about them. Not the mouse. Make it mic off කරන්නකෝ. මේකේ speakers off කරන්නකෝ. Speakers off කරන මට මෙතන දැන් control කරගන්න බෑනේ ඔයා ඔහොම කරලා දුන්නා. වැඩක් නැහැ නේ මේක හැදුවට. ඒ වුණා මට control එකක් නැහැ. Right, people, photosynthesis. So photosynthesis, 2 steps, the light light dependent reaction. And what is the second one? Kelvin cycle, right? Light dependent reaction and Kelvin cycle. Now people, the first one, light dependent reaction. Now light dependent reaction, we talked about this. These photosystems. There are two photosystems available. There are two photosystems. What are those two photosystems? We say, people, photosystem 1 and photosystem 2, right? Photosystem 1 and photosystem 2. So as light strikes, we learn that the energy is going to share among these pigment molecules like this. And finally, people, energy arrives at P680. Finally, energy arrives at P680. Can you tell me the structure of photosystem? Structure of a photosystem, important part. Can you tell me the structure of the photosystem? Photosystem had two parts. It had two parts. One is a light harvesting complex. So somewhere here, a light harvesting complex with some pigments, right? And then we said that the center complex was there, center complex. And then there were two pigments in the center complex, two special pigments, P pigment. We said P680 and P700 in different systems. You learn that. So when light strikes like this, the energy share, and finally energy reaches here at the wavelength of 680 nanometers. And then people, some complex P prime EA, PEA. What is PEA? Right? What is PEA, Santosh? What is PEA? Primary electron acceptor, right? Primary electron acceptor, right? So from there, the electrons are going to direct into people's series of electron carriers, right? So we talked about this electron carrier series. Series of electron carriers are there. So these energy reaches here and passes along these electron transport chain. They said that people, the energy ATP forms here. And then that energy production is an indirect process. We learn, people, the actual process behind this, the ATP production. But anyway, ATP forms. These electrons finally reaches people P700. They reaches to P700. And in the meantime, we learn, people, the P700 photosystem, that is photosystem 1. Photosystem 1 receive light energy. Photosystem 1 receive light energy too. And photosystem 1, by receiving light energy, we can see, people, it also emits electrons. So we can show it like this. This is one photosystem, primary electron acceptor. It is part of the center complex. And then here we have another primary electron acceptor. And then it's a part of another center complex, right? So we learn that when the light hits here, light hits this one simultaneously. And then originally we learn, people, the photosystems, the photosynthetic pigments, and these photosystem 2, right? This is photosystem 2, PS2. PS2 emit electrons. How many electrons? 2 electrons. They pass through this electron transport chain, series of electron transport chains like this, making energy ATP here. These electrons are going to flow through this. Electrons are going to arrive here, right? Electrons are going to arrive here. But in the meantime, what happens? As this one receives energy by light, and as these photosystems transfer that energy. Finally, here, this is going to emit electrons. Ne? 2 electrons are going to emitted from here. These electrons are finally receiving people for an enzyme. Remember the name of the enzyme? What do you call that enzyme, people? What do you call the enzyme? What do you call this enzyme? This is NADP plus reductase. Ne? Reductase. NADP plus reductase. So it has a job. Ne? That's job is to form NADPH from NADP plus. By taking some H plus, it takes H plus. It takes H plus and then forms NADPH, right? So that is how actually the energy NADPH is going to be formed. So there are two products. Ne? One is ATP, the other one is NADPH. Two products are going to form in this sequence of events. So if you take people, two products are there. Eh? But you know, people, this cycle can't run without water. So somewhere here, water breaks and then releases half oxygen. Eh? Releases oxygen. This donates people electrons, 2 electrons to, what do you call, reduce the oxidized center complex pigments. Now you learn here the P pigment P680, 2 of them available which undergo ionization, 2 molecules of P680 plus it emit electrons, right? So somebody must donate electron here for this to reverse the process back to P680. The electron donor is water. Water provides the required electrons. And then people, it provides the required electrons for this process to happen, right? It donates electrons for this process to happen, right? And so that is the process. So that the electron donor is this. So who is the final electron acceptor? NADPH. You learn, people, the electrons are flowing starting from water on this path and ending up with NADPH. What do you call that process? 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 Which electron flow is that? Which electron flow is that? Lean electron flow. And then you learn, people, that there is another electron flow process here. Cyclic electron flow is also there. So those are things we learn for the light reaction. And then we will take a piece of paper. Can you write Kelvin cycle with your memory? See if you can write the Kelvin cycle steps. That's the second part. Second part, Kelvin cycle. So this is the light dependent reaction. Light dependent reaction. See if they can write Kelvin cycle. See if they can write the Kelvin cycle. Right. So, oops. We talked about this cyclic electron flow. Cyclic electron flow now. And then we have to talk about people Kelvin cycle. You know people, Kelvin cycle has 3 steps. What are those 3 steps of Kelvin cycle? 3 steps. Number 1, this one, carboxylation. Number 2, reduction. Number 3, regeneration and regeneration of carbon dioxide acceptor. So REBP is the acceptor there. So the REBP acceptor is going to reform. REBP acceptor is going to reform. So 3 steps, right? 3 steps. You have to write that. How do you write the Kelvin cycle? Kelvin cycle. Most of the time, people, when I ask in a revision class, students who write the Kelvin cycle, they all write Kelvin cycle. In a second, people, the moment I ask, they draw the Kelvin cycle. What they draw is just this one, right? And sometimes people, as extra information, they write Kelvin cycle, right? Is it the same thing that you did now? Did you write the complete cycle, or is it just Calvin cycle? This one is Calvin cycle. So you have to tell me the steps now. Something available here. Come on. What's that? Come on. What should I write here? Right. What should I write here? RUBP. That Santosh RUBP. RUBP. That's correct. Good. How many carbon, Santosh? 5 carbon. The 5 carbon. And then carbon can join carbon dioxide. Come and join here. And then people forms unstable C6 compound. The enzyme is Rubisco. Rubisco, and forms an unstable C6 compound. Unstable C6 compound. What is the stable compound which forms? For stable compound, what is that? That is 3 molecules of PGA, 3 molecules of PGA. How many carbon in each PGA? 3 carbon. Right? And then that 3 carbon transforms into another intermediate, right? Another intermediate which transforms into G3P later. The G3P later. So what is that intermediate? What is that intermediate? What is that intermediate? What's that? What's that? Come on. Come on. What is it? What is that? Come on, tell me. 1, 3. I'll say 1, 3. 1, 3, biscuit. 1, 3, phosphoglycerate. 1, 3, bisphosphoglycerate. 1, 3, bisphosphoglycerate. And then people, PGA to 1, 3, bisphosphoglycerate. Something involved here. PGA has 1 phosphate, 1, 3 has 2 phosphate. So a phosphate donor, ATP becomes ADP. Now, people, to balance this, we have to take 6 RUBP. 6 RUBP, no, 3. We took 3 RUBP, 3 carbon dioxide, and then here, how many PGA? 6 PGA, so 6 ATP, 6 ADP, 6 of this forming 6 G3P, 6 G3P. Then 1, 3 bisphosphoglycerate becoming G3P, people. What involves here? NADPH becomes NADP plus, right? NADP plus, and then forms G3P. G3P 6, 1 goes out. 1 G3P goes out. 5 G3P returns, and then becomes triple RUBP back again. Certain other information about the phosphate elimination, those things we learn here, people. I'm not going to talk about that. But anyway, there are 3 steps here. Calvin cycle has 3 steps. From here to here, number 1. From there to here, number 2. From this point to back to RUBP, number 3. So number 1 is carboxylation or carbon fixation. Number 2, people, reduction. And then number 3, people, we take as the regeneration of RUBP. Regeneration of RUBP. The question is based on that, the process. So you learn this. We talked about it, people. Carbon fixation. The carbon dioxide receptor is 5 carbon sugar molecule, ribulose bisphosphate. The addition of carbon dioxide RUBP is called as carboxylation. The enzyme involved in this process is RUBP carboxylase oxygenase or Rubisco. And then the first product of RUBP carboxylation is 6 carbon molecule, unstable, and it breaks, people. And then 3 phosphoglycerate forms. And then this is the statement in question. This statement is little in question, people. They have taken it here. 3 PGA is converted to 1, 3 bisphosphoglycerate by adding 1 phosphate group from ATP. That they have taken for carboxylation. Fine. right? This 3-PGA is converted to 1,3-bisosylglycerate by adding 1 phosphate from ATP. That's correct. This is perfect. That's all right. But then here we have another problem. What is that? Problem is 1,3-bisosylglycerate will be reduced to glyceraldehyde 3-phosphate through step-by-step enzyme catalyzed reaction using NADPH and ATP. Now, this is the problem. There aren't any 2 ATP usage here, right? There aren't 2 ATP usage. It's just 1 ATP molecule here. And those ATP involved here, they are like have mentioned it here also. That is a weakness in the resource book, right? We can't describe it. So when explaining that, can we draw this diagram? No, you should not draw the diagram, because if something is not given in the resource book, they're not going to expect that as a diagram or a chart. So no need to do that, right? Simply explain it. Simply explain it. And then people, regeneration of REBP. Regeneration of REBP. REBP is regenerated. So that's the event. Then we learn people about, then we learn about the photorespiration. Photorespiration. Now we learn people, some plants are having special process. What is that? C4 photosynthesis. Why plants have C4 photosynthesis? There's a problem with rubisco. Rubisco enzyme is a troublemaker. It's not a genuine partner. That's the problem. When there is someone, when there is a husband, not genuine, having different relationships. That's the same problem with this poor rubisco, right? RUBP plus carbon dioxide is giving people 2-PGA. That is what we expect for the rubisco. That is what we expect from rubisco. Rubisco, right? That is what we expect from rubisco. But rubisco is not genuine. Why? Rubisco like to react with carbon dioxide and REBP. Actually, REBP is a problem with REBP, not exactly with rubisco, right? REBP is problem. REBP like to react with carbon dioxide, right? But REBP also can react in absence of carbon dioxide. In absence of carbon dioxide, REBP react with oxygen and form 1-PGA. 1-PGA and 2 carbon product, which is toxic. 2 carbon product, which is toxic. It's done by the same rubisco. It's done by the same rubisco. So in normal condition, when the atmosphere has enough carbon dioxide, what is the atmospheric carbon dioxide level? 0.04. We took it as 0.04 carbon dioxide in the atmosphere, right? In a hot sunny day, we learn a problem. In a hot sunny day, plants have a problem. Plants have a problem in hot sunny day. Right? Plants have a problem in a hot sunny day. What is that? They lose too much of water from their leaves. Too much of water they lose from their leaves. And special people through these tiny pores present on the leaf. Tiny pores on the leaves, they lost a lot of water. So they have to close their stomata. When they close stomata, they begin a new problem. They don't receive enough carbon dioxide, and then oxygen trap more inside. Now the rubisco is happy. Why? He get the oxygen. Rubisco now don't receive carbon dioxide, instead receive oxygen. So it carries out this process and form 2 carbon product. You learn people this 2 carbon product is toxic for the plant. So it need to be removed immediately. So this 2 carbon product is going to transform into it's going to transform into PGA back again, plus carbon dioxide is going to be eliminated, right? So this process is not easy. And then you learn people, 3 organelles are going to participate. CHP means chloroplast, mito means mitochondria, peroxisome, actual organelle, the order is mitochondria has to come last, mitochondria, right? Chloroplast to peroxisome to mitochondria. These and then back again, people from mitochondrion to peroxisome to chloroplast. This PGA, this 2-carbon product flow, acid flow, PGA is going to reform. So if I show you it like this, this is chloroplast. This is that peroxisome. This is the mitochondrion. This is the mitochondrion. So what happened? The 2-carbon product, it goes to peroxisome. It goes to mitochondria. Inside that, it converts and then come back to the peroxisome and then come back as PGA, back to the chloroplast. So it's like moving down and then returning. So it's a process. You don't have to know the details about it. But what happened? The 2-carbon product is now transforming into many intermediates, and finally people transform back into PGA. Not one 2-carbon product, right? We said people need how many? 3 of them. 3 of them can form 2 of them, PGA. 3 of them is 6 carbon, and then form 2 of them with 3 carbon. So this process is something like that. But anyway, people, there is a carbon loss also. So this balancing is not fair. I said that 3 to 2 just to show that it can be produced. But then some steps are there with some carbon dioxide elimination here, with some carbon dioxide elimination there. This event can happen. So finally, PGA is going to reform. But you understand, people, there is a significant energy involvement here, and part of carbon dioxide is going to be lost also. So this process, the photorespiration, as it happens, the productivity decreases. So therefore, people, this reaction, as it happens, less PGA is produced, almost half, so that the reactivity decreases. So that's the problem with the C3 plants. They carry out this event, what do you call, people? Photorespiration. This event we call as photorespiration. Why respiration? Why photo? Because it happens only in bright light condition. And why it is respiration? Because oxygen involved. And this is a sugar also, ribulose, bisphosphate. It's a phosphate containing sugar. Then you can see that, people, the oxygen involved sugar breakdown, something similar to cellular respiration. So this happens. And then, people, we learn the process prevents by a new pathway, evolution of new pathway. So can you please revise cellular respiration too? Yeah, sure. But today we have RK, respiratory quotient. I think I can. Yeah, sure. Right. Then cell cycle you have to study then. Cell cycle and enzyme part, then you have to study, right? If I go into that. So this process is now to prevent that, the plant have evolved, people, new photosynthetic mechanism. New photosynthetic mechanism. What is that photosynthetic mechanism? C4 photosynthesis. C4 photosynthesis. So we talk about it. We talk about C4 photosynthesis. Let me have a small drink. Cheers. Cheers. Right. C4 photosynthesis. Now, people, in C4 photosynthesis, we learned that the photosynthesis happen in 2. We learned that the plant has differences too. So, dear sir, kind of request to upload the revision tute to the revision class course in the website. Yeah, sure. There's a small error in the website. It didn't allow me to upload. I'll upload it tonight. Now we have fixed it. We can do that. Only 2 tutes are there. You need 4. Oh, yeah. I'll upload them. A revision student in this class, this is for babies, right? 2027 babies anyway. Right. Glad that you stay here. Right? It's okay. So please revise cell cycle or no time today to do everything because this is just 1 hour session, 1 hour 50 minute session. So will it be a problem if we do one class physical and one other online? I think this is in my schedule at the moment, right? This is in the schedule. Can't go out of the schedule anyway. Right. So C4 part, people will quickly check this C4 photosynthesis, Lamai. C4 photosynthesis, you learn the process. You learn some differences between these two plants. We learn the differences between these two plants. When it is a C3 plant, if you take a leaf cross section, you take the leaf cross section, you can see people in the leaf cross section. Let me draw it here. Let me draw it here. This is a C3 plant leaf cross section. C3 plant leaf cross section. And here we can see people, the upper epidermis. Upper epidermis, this is the vascular bundle. And then people, this is the lower epidermis. This is lower epidermis. And then here we can see only mesophyll cells are there. Mesophyll cells present. Right? So you take mesophyll cell. You take this mesophyll cell. This mesophyll cell has chloroplast. Mesophyll cell has chloroplast. It has a chloroplast like that. So all events happen inside this chloroplast. All events happen in this chloroplast. Right? What I meant by all events mean the Kelvin cycle as well as the light dependent reaction. So here the light dependent reaction, LDR, happen here on the thylakoid membrane. LDR occur on the thylakoid membrane. That's like a special feature, right? This is light dependent reaction, LDR, happen on the thylakoid membrane, and then people in the stroma, the Kelvin cycle occur. Kelvin cycle occur in the stroma. So both events happen. People in the same area. Both events happen in the same area, same chloroplast, right? But when come to a C4 plant, then that's different. In a C4 plant, it's different. What is the difference in a C4 plant? If I take people C4 plant, if I take a C4 plant cross section like this, if I take a C4 plant cross section, right? And then if you take it cross section like this, if you take a cross section like this, if you take the cross section of the C4 plant, we know people tightly packed cells present around them, tightly packed cells present. And you see people, Kranz anatomy is available. The bundle sheath, the vascular bundles are covered by a bundle sheath. How do you say Kranz anatomy? Kranz anatomy. Kranz anatomy. Kranz anatomy present. What we said, Kranz anatomy, vascular bundles are, Kranz anatomy, vascular bundles are surrounded by cells. and that is surrounded by some more cells. That sort of arrangement is the Kranz anatomy, right? And then here, people, if you take there are two cell types with chloroplast. Chloroplast is available in these mesophyll cells as well as in the bundle sheath cell, as well as in the bundle sheath cell. You don't see that in C3 plant. In a C3 plant like this, chloroplasts are only present in the mesophyll cells. Chloroplasts are only present in the mesophyll cells. They don't have chloroplasts in the bundle sheath. Chloroplasts are not present in the bundle sheath. But that is not the case. That is not the case when you're talking about a C4 plant. C4 plant, that's not the case, right? So when we enlarge the arrangement, when we enlarge this structure, I'll go to a new page. You can see those two cells. You can see those two cells like this. So how do you say that? The cycle begins with people PEP, PEP, PEP converts to OAA with the carbon dioxide, which comes from outside. With carbon dioxide comes from outside. Carbon dioxide, with carbon dioxide which comes from outside. Carbon dioxide that comes from outside. OAA forms. OAA become malate. O aspartate. Malate move into the bundle sheaths. Now this is mesophyll cell. Mesophyll cell. This one is the bundle sheath. Bundle sheath cell. So they move, the malate move in, and then malate convert into pyruvate. Pyruvate, which is a 3-carbon containing compound. So malate is a C4 carbon containing compound. This process is going to release carbon dioxide. This carbon dioxide enters into the C3 cycle. This carbon dioxide enters into C3 cycle. This carbon dioxide enters into C3 cycle. What do you call people? Calvin cycle. This carbon dioxide goes to Calvin cycle. This carbon dioxide goes to Calvin cycle. And then the pyruvate return. And then transform back into PEP. So there is an extra process. Now, the normal Calvin cycle run here. The normal Calvin cycle run here. Normal Calvin cycle run here. But there is an extra cycle, this extra process. So when they ask for you to write the C4 pathway, this extra part as well as the Calvin cycle you have to describe. Extra part as well as Calvin cycle you have to describe, right? As this process, right? Extra part as well as the Calvin cycle you have to describe, right? So that is what you call the C4 photosynthesis. I think this is enough. C4 photosynthesis. You learn people some differences between. C4 pathway and C3. Differences you learn here. C3 plants and C4 plants, differences you learn. What differences you learn, people? What plants are showing that? Wheat and rice, barley, people, C3 plants. Maize, sugarcane and grasses are C4 plants. Then we learn, people, carbon dioxide fixation. Carbon dioxide fixation occur once. Here occur twice. Right? First, carbon dioxide is fixed by PEP. Secondly, it's fixed by REBP. And then people, carbon dioxide acceptase is only REBP. This is PEP and REBP both. And then carbon dioxide fixing enzyme, if you take people, Rubisco is the enzyme here, P carboxylase as well as Rubisco, both are involved there. And then first product is a C3 acid, that is PGA, and C4 acid oxaloacetate is the first carbon, first stable product of photosynthesis, right, people? And then here leaf anatomy, bundle sheath cells and mesophyll cells present. Kranz anatomy is available, Kranz anatomy present, right? Kranz anatomy is there. So some differences are there. Some differences are there. Those are the things we learn, people here, photosynthesis. So I want you to go through that. I want you to go through that photosynthesis part quickly once again. You learn some significances in photosynthesis, some importance in photosynthesis. What did you learn, people? We learned that the significances of photosynthesis, these points, significances of photosynthesis. Where was that? Significances. Significances of photosynthesis. These things we discussed. Where is it? Right. We learn, people, I think, significances of photosynthesis. Help the plant to improve the efficiency of carbon dioxide fixation at lower carbon dioxide level. And then people, carbon dioxide can be concentrated effectively. So in hot, dry condition, they can close stoma even. They can close stoma and they can maintain their photosynthesis when it is a C4 plant. Better water usage efficiency is there because they lose less water because they can do the photosynthesis even with a partial closure of their stomata. And then Rubisco is very efficient in function, so therefore need less Rubisco. That means the available nitrogen can be saved without wasting unnecessarily. If you have some machine which is, if you have a light bulb, they get too much heated and then produce people less amount of light, and then you lose a lot of electricity. Same like that. Rubisco is very efficiently functioning, right? And in a C4 plant compared to a C3 plant, right? So that needs, so the plant need less number of Rubisco, so that saves some extra amount of nitrogen for the plant. All right, people, right? Then people will move to talk about cellular respiration. Is it here in the paper? Practical part is there. Practical part is there. Practical part, right? We learn about cellular respiration. How do you define process by which chemical energy in organic molecules such as carbohydrates released by stepwise oxidative process? Stepwise oxidative process released. We learn, people, respiration can be aerobic respiration and anaerobic respiration, but you learn, people, you learn in cellular respiration, you learn glucose. Glucose turn into pyruvate, and then pyruvate enters into the mitochondrion. Enters into mitochondrion. Now you have to understand, people, the prokaryote and the eukaryote. Prokaryote and eukaryote. The prokaryotes don't have mitochondria, but instead they have mesosomes. You learn about mesosomes, some membrane foldings are there. Prokaryotic eukaryotic cell comparison, we learn that mesosomes are there. And then they use these mesosomes, people, for the, they use mesosomes for the cellular respiration process, so that every time, people, when photosynthesis occur, every time when cellular respiration occur, there is no requirement to have mitochondria. Really? Why is it raining heavily? Romina, is it raining heavily in your area? Worrying, ne? Because another cyclone, not a cyclone yet, ne? Is a tropical depression is on the way. Is it raining heavily? It's not raining here. Very calm and quiet. Dead silent. Not even a leaf move. That's dangerous, ne? Right? Every time before a storm, it happens. Raining. Now came raining. Okay. So here, people, the prokaryotic cell, we'll divide it. Let's see how we can divide it. Cellular respiration. Cellular respiration we can divide as aerobic and anaerobic. Aerobic respiration has glycolysis and pyruvate oxidation, and Krebs cycle. Krebs cycle. And then electron transport chain, that is oxidative phosphorylation, right? Anaerobic respiration also has two steps, glycolysis. So it's a common event for both glycolysis and we can say people two branches here, ethanol, ethyl alcohol, ethyl alcohol fermentation, and then second one we can call as the lactic acid fermentation. Lactic acid fermentation, right? Now you have to understand here, the aerobic respiration can happen in prokaryotes plus eukaryotes, prokaryotes and eukaryotes. It can happen, right? If prokaryotes, glycolysis occur in cytosol. Krebs cycle is also in cytosol. Then electron transport chain in prokaryotes happen in mesosome. I'll use red color for prokaryotes. Anaerobic respiration in prokaryotes, glycolysis in cytosol. Sometimes people in prokaryotes, ethyl alcohol fermentation or lactic acid fermentation can happen. That also happen in cytosol. Prokaryotes can carry out ethyl alcohol fermentation. Now, bacteria can carry out ethyl alcohol fermentation as well as lactic acid fermentation. We learn, people, most of the bacteria carry out ethyl alcohol fermentation, and then yeast. Examples, yeast is an eukaryote. And then lactic acid fermentation is done by bacteria in yogurt and curd. That is what the resource book says. Then people, so here, eukaryotic ones, people, glycolysis occur in cytosol. And then eukaryotic ones, the pyruvate oxidation and the Krebs cycle occur in mitochondrial matrix. And then here people, electron transport chain occur in eukaryotes where cristae, right? In prokaryote, glycolysis is in cytosol. Sorry, eukaryotes, glycolysis in cytosol. And ethyl alcohol fermentation, and that one happen in cytosol too. Right? That happens in cytosol. So we discuss these things, right? And we discuss these things, right? Hope this is clear for you. Hope this is clear. Now people, cellular respiration Cell respiration, glycolysis. We'll take the first one, glycolysis. Glucose transform into 3 carbon containing G3P, 2 molecules of G3P from 1 glucose, 2 molecules of G3P and 2 molecules of pyruvate. 2 molecules of pyruvate. Here initially 2 ATP becomes 2 ADP. And G3P to pyruvate people, what happened? 4 ADP forms a 4 ATP. So you write the overall reaction. Here what happened? 2 ADP becomes 2 ATP. That's the net productivity. That is the net productivity. So we take it as net. There is 2 net ATP production. In addition to that, here we can see that people 2 NAD plus becomes 2 NADH. 2 NAD plus becomes 2 NADH. Right? 2 NAD plus becomes 2 NADH. So you know people, these NADH, 2 of them form. Right? 2 of them form. Then people, pyruvate. Then you learn this event. Then pyruvate enters into the mitochondrion. This is that mitochondrial membrane. This is the mitochondrial membrane. That's the mitochondrial membrane. Outer membrane, inner membrane, intermembrane space. So this pyruvate enters into the mitochondrion, 2 pyruvate. 2 pyruvate enters into the mitochondrion, and inside the mitochondrion, this becomes 2 acetyl. Acetyl has 2 carbon. Pyruvate has 3 carbon. Pyruvate has 3 carbon. G3P has 3 carbon. Glucose has 6 carbon. Pyruvate has 3 carbon. 3 carbon. 3 carbon. This is 2 carbon. Right? Acetyl. And then people, it combine with something called coenzyme A, right people? CoA. Coupled with CoA. This reaction is a decarboxylation. So carbon dioxide leave. Carbon dioxide leave. 2 carbon dioxide leave. From every pyruvate, 1 carbon dioxide. From 2 carbon dioxide. And then form acetyl. Then the acetyl enters. Into the Krebs cycle. Acetyl enters into the Krebs cycle. One more important thing. Here we saw that 2 NAD plus becomes 2 NADH. The process is going to suck. Coenzyme A. And then as it form, what it forms will acetyl coenzyme A is going to form. And then acetyl coenzyme A undergo this sort of a cyclic process where 4 carbon containing OAA. How many carbon? 4 carbon, OAA. Together with the 2 carbon in acetyl, it becomes people citrate. Citrate. How many carbon in citrate? 6 carbon in citrate. Right? 6 carbon in citrate. It becomes people 5 carbon. It becomes 4 carbon. It becomes another 4 carbon compound. 4 carbon. 4 carbon. So series of 4 carbon containing compounds are going to be generated here. Series of 4 carbon containing compounds are going to form. Right? Series of 4 carbon containing compounds. Now the easier part is this. 1 NAD plus NADH form here from NAD plus. Right? And then people, another same event happen here. NAD plus becomes NADH. Then here people 1 ATP, 1 ATP, ADP becomes ATP. I'm talking for every acetyl. right? And then here people, FADH2 forms FADH2 forms from FAD, and then the last one, another NADH from NAD plus, right? Those are the events. This is slightly different because there is another 4 carbon intermediate. Forget about it. So this is what do you call, people? This is the Krebs cycle for every acetyl. Now we have 2 acetyl. So then we have to multiply everything by 2. The cycle now run 2 times. So this is 2. This is 2. This is 2. This is also 2. ATP, 2 ADP, 2 ATP, 2 FAD, 2 FADH, 2 NAD plus 2. That's it. Krebs cycle. This is Krebs cycle. Krebs cycle. So, Krebs cycle. 2 times. Krebs cycle occur now 2 times. Right? Krebs cycle occur 2 times. That's it. That's it. Now, all these things have to feed people into the electron transport chain, which happen here on the inner membrane of mitochondrion. On the inner membrane of mitochondrion, a special event occurs, electron transport chain, right? When FADH2, when NADH is fed to this process, it becomes NAD plus, right? This NADH can enter into this electron transport chain. One NADH enters and then return back to NAD plus. Then 2 electrons are going to be released. 2 electrons are going to be released. As the 2 electrons are passing, ATP form. ATP form. How many ATP? 2.5 ATP form for every electron pair given by NADH. 2.5. How many NADH? 2, 4, 6, 8, 10 NADH. For one glucose molecule, 10 NADH forms. Every NADH, 2.5 ATP. So how many NADH? 10. So how many ATP can be produced, people? How many ATP can be produced? 25. That's it. That's it, 25. As it flow along this electron transport chain, these electrons are accepted by people, molecule oxygen. Oxygen molecule is the final electron acceptor. What it forms is water. It forms water. So when oxygen present, this reaction of ATP production can happen. That is why we call it as aerobic respiration. Oxygen needed. Oxygen is a final electron acceptor. Final electron acceptor is oxygen. So the event can happen. Now the special thing, every FADH2 can form only 1.5 ATP. So how many FADH2 form in the process? Only 2. Now NADH 10 from the glycolysis, and then people from pyruvate oxidation to acetyl, everything 10 NADH can be produced. But from this entire process, the maximum number of FADH2 is just 2 FADH. Only 2. Every FADH 1.5 ATP. How many ATP then? Maximum 3. So 25 plus 3. How many? 28 ATP can be produced by the electron transport chain. But you understand there are 2 ATP available here. There are another 2 ATP available there. So that means 28, you have to add another 4 ATP out of, what is the total then? What is the total then? 28 plus 4, 32. That 32 ATP production happen in liver cells and liver cells and what is the other one? Liver cells and what is the other? What is the other? What's the other one? Liver cell and what's the other? What's the other? Cardiac cell. Heart muscle cells. Heart muscle cells. That's correct. Ne? Now, in other cells, any other cells, this pyruvate transport needs some extra ATP. 2 extra needed. Ne? Liver cells are efficient. They don't need extra ATP. Other cells need extra ATP. So, for other cells, the total ATP production is 30 ATP. For liver and the cardiac muscle cells, the total ATP production is 32. 32 ATP can be produced. And that's the aerobic respiration part, my dear. Now we move to anaerobic respiration. Where this pyruvate transform into lactate, lactate or lactic acid, or else it can become acetaldehyde, acetaldehyde, and later turn into ethanol. The whole purpose of this anaerobic respiration is to convert this NADH into NAD+. The whole purpose is that when oxygen is unavailable, right, what is these electrons are not going to be accepted by anyone. So whole electron transport chain is going to fail. Whole electron transport chain fails, right? So therefore the entire Krebs cycle fail. Why? Without NAD+, because the electron transport chain converts NAD+ into NADH. So that the whole Krebs cycle and the pyruvate oxidation fail when oxygen is absent. So what happened, people? Pyruvate can't even enter into the mitochondria. So what happened now, people? Anaerobic respiration can maintain. You know, anaerobic respiration has 2 steps. The glycolysis and the fermentation. That even going to fail, people, if this NADH is not cycled back into NAD+. Do you understand that? This is important. So that conversion is done by converting pyruvate into lactate, people. This H+ is absorbed into pyruvate. So we say that the hydrogen acceptor is pyruvate. This H+ is absorbed into pyruvate and pyruvate become lactate. And NADH now become NAD+. So the glycolysis can maintain because NAD+ is available. NAD+ is recycled. So the whole purpose of anaerobic respiration, is to cycle which one, people? NAD+. NADH to convert that into NAD+. You understand that? That's the whole purpose. So it happens here. That happens here. That happens even here. That happens even there. NADH become NAD+. Right? So, but here, you know, people, this 3-carbon pyruvate first become 2-carbon acetaldehyde. 2-carbon acetaldehyde with a decarboxylation. So in ethanol fermentation, NADH is not absorbed into pyruvate, but NADH hydrogen is absorbed into acetyl. NADH hydrogen is absorbed into what, people? Acetyl. And then transform into ethanol. So final hydrogen acceptor is acetaldehyde. Final hydrogen acceptor is acetaldehyde. Right? Understood? Clear? Harida? Right? So you learn, people, lactic acid fermentation is done by some curd and bacteria involved in curd formation. Right. We discussed this very recently. Lactic acid fermentation. What we say, people, what are the microorganisms in here? Lactic acid fermentation. They say certain fungi and curd and yogurt forming bacteria. Then ethanol fermentation, they say many bacteria and yeast, many bacteria and yeast. So yeast is eukaryote, the other one is a prokaryote. Right. I think that's clear. Now, people, let's talk about our respiration experiment. RQ respiration experiment to measure RQ. How do you measure R cube. Respirometer, using a respirometer. We use a respirometer. And how do you measure the respiratory quotient? R cube. R cube is carbon dioxide volume. Ne? R cube, how do you write? Carbon dioxide volume. Volume of carbon dioxide release to volume of oxygen absorbed. Ne? That is R cube. So we have this setup. We have this setup. This is the setup. This is the setup. Ne? This is the setup. You place people KOH containing tube inside. Seeds, how many grams? 25 grams of seeds. Water bath. You keep this in a water bath to maintain the temperature. Because the germinating seeds can heat up. And then you have a color liquid here. Colored liquid in this part. Colored liquid. So you set it up. And then you close this tap. Set it up. Balance this AB liquid columns by opening the tap. This is now air sealed system. You apply some Vaseline or clay and then air seal it. Then you open the tap, balance the two liquid columns and close the tap. Then the two liquid columns are equal. This column goes down. Starting from this zero line, it goes down because these seeds absorb oxygen. Seed absorb oxygen. And then they release carbon dioxide. This released carbon dioxide is absorbed into KOH. Released carbon dioxide is absorbed completely into KOH. That is what we assume. So you take people 100 rupees from somebody, and then you give him 100 rupees back again. As you try to give, some other friend is going to seize it from you. And then you owe now 100 rupees. Same thing happening. It takes oxygen and give out carbon dioxide. Sometimes you take 100 rupees and you give some money. It's not exactly the same 100 rupees you give. Some money you try to give, and that money is now taken by KOH. So what happened, people? Sometimes the absorbed oxygen and the released carbon dioxide level is equal. Now, for an example, if carbohydrate containing seeds does this, these two volumes are equal, right? Sometimes in lipids, people, more oxygen is absorbed and less is given, so that it's not equal. Protein also same, more oxygen absorbed and less carbon dioxide is given out. So we can find that difference. So oxygen is absorbed, release carbon dioxide also absorbed. So due to the oxygen absorption, there is a volume drop. Due to oxygen absorption, volume drop. If carbon dioxide release is there, volume is going to increase, right? Let's take, this is dropping, and due to carbon dioxide release, it goes up once again. So this is the overall change. You understand that, right? But now what happened? It's dropped due to oxygen. The released carbon dioxide get absorbed into KOH. So it doesn't go up because it's absorbed now. So as a result, this volume is, this is the volume of oxygen. This is volume of oxygen. So with KOH, you get volume of oxygen. You get volume of oxygen. Now you do something. You remove KOH from the tube and do the experiment once again. You do the experiment once again by removing. Now what happens? Same amount of oxygen is now absorbed, and then the column goes down. And then the column return, carbon dioxide now return. This is volume of carbon dioxide, right? So it's now going to stop somewhere here. This is volume without KOH, right? This is volume with KOH. Let's take that, people. This is 0. This is 100, right? Let's take this this position as 70 kilo, 0 to 70. Then what is the carbon dioxide level? How do you find that? You get the difference. This is 100 and you deduct 70, you get this one. So you can find carbon dioxide volume. You can find volume of carbon dioxide. How do you find volume of carbon dioxide? Let's take that, people. This one as H1. This is H1 and we'll take this one as H2. So volume of carbon dioxide is equal H1 minus H2. right? H1 minus H2. So we can apply it here. R cube, H1 minus H2, and oxygen volume is H1. So you can find R cube value. This R cube value tells you people what sort of the respiratory substrate is used by the seed. If it is a mung seed containing carbohydrate, R cube value is, what do we say? R cube value is 1. It has carbohydrate. Then some resinous seed, some oil containing seed, the R cube value is going to be 0.7. More protein containing seed, this can be around people 0.8, right? So it's variable like that. I hope this is clear. Then people, that's all for the day discussion. Right? Thank you very much for your participation. I can take further longer. I know you, right? You can't watch continuously the screen. I don't want you to do that. So this is enough. So you do the paper number 4. I have uploaded it to the website. Revision papers, if somebody wants, I'll upload them tonight. Now, soon after this class, I'll upload it. Then thank you very much. Thank you very much. And we're going to meet people once again soon. Right? Saturday. Hopefully on Saturday, because this the weather condition is going to be people little upset tomorrow. Right? Tonight and tomorrow. That's their maximum prediction. Maybe it will die out, hopefully. And if not, people tomorrow will be little heavy rainfall. So we'll meet on Saturday. Right? Thank you, and big salute for all of you who stayed over this time period. I'll meet you again. Bye. Good night.